for the introduction. It's really cool to be at ISMB this year. So yes, today's talk is about sequence-to-graph alignment. And this is joint work with my PhD supervisor, Veli Makinen, and our colleague, Manuel Cáceres, also from Helsinki University. And yeah, this was funded by Alpaca, HeAT Institute, and also Timpermed. And so yeah, today I'm talking about graph pangenomics, right? And in Tobias's talk this morning, I hope he convinced you that graphs are cool and that they can represent variations compactly. And indeed, when I talk about graphs, I mean these sequence graphs where the nodes are sequences, the edges are connections that are supported by some of the haplotypes, and the paths in the graph, they actually spell the genomes that we want to study. And as a result, the sequence-to-graph alignment emerged as a central problem, and specifically for long-read alignment, the practical aligners, the state-of-the-art aligners, use these seed cluster extend and seed chain extend heuristics, which consist of these 3 steps, which is seeding, so finding good matches between the reads and the graph, and then this optional chaining step that filters the reads into good chains that are compatible with an alignment, and then finally extending into a full alignment at the base level. And indeed, there are many different sequence-to-graph aligners, and they're all very cool in their own specific application, but also they solve the same problem, so they can be compared with one another. And yeah, these aligners are graph aligner, mini-graph, graph chainer, mini-chain, and this is an active area of research, and indeed, VG-Giraffe is a short-read aligner, but they published a long-read module this year. And picking graphs as the object where we want to align also brings some challenges. Indeed, many different graphs can represent the same input sequences, and the HPRC human graph references are built with 3 different construction methods. And each of these graphs implies also a recombination model in how the paths can recombine actually in the graph, and as specified by the connections in the graph. And matching a pattern exactly, so exact pattern matching in graphs is actually a really hard problem. It's as hard as actually aligning to the graph, which is not the case in the sequence-to-sequence case. And this actually limits the options that one can have when you are doing seeding, which is the first important step. But nonetheless, chaining techniques were approximated and extended to graphs, and also the alignment techniques were extended to graphs. But the state-of-the-art for long reads remains heuristic and minimizer based on minimizers, seeds. And our result is that we present a complete workflow based on indexable elastic founder graphs and test it at the scale of a human chromosome. And elastic founder graphs, they actually simplify the pangenome construction because they start from a multiple sequence alignment. So we take that as the truth. And then we segment this multiple sequence alignment into a graph. And the way of making this graph indexable actually is a strength because we have a very conservative approach to recombination. So we allow recombination only after having read a unique sequence. I will explain this in detail later, but for example, here there is recombination between the input haplotypes because this letter C occurs only starting from that column of the MSA and nowhere else. So it's a unique sequence. And the overview of the workflow is that we will have a very expensive part at the start, which is still the construction. So we can start from a BCF file, so reference plus variations, and we convert it into a multiple sequence alignment. And then we can actually find all these unique regions and then segment the graph into an indexable EFG. And from here, I will try to show that the benefits are that we actually, with a pattern matching index, we can actually get fast and accurate alignment. And all these tools are implemented as specific parts, independent parts of the toolchain. And so we implemented the seeding with EFG locate, chaining with chain X block graph, and we actually didn't implement the extend part because we can use the graph aligner extension algorithm. And just to get more details about elastic founder graphs and how we can build them out of short and unique sequences. So as I said, they are variations graphs obtained from the segmentation of aligned sequences. So what happens in each of these segments after you pick the segment is that you just merge the unique sequences. And if 2 sequences diverge, then they will be represented as 2 different nodes. And this indexable property that we require is that all node sequences can only be can be spelled only starting from one position only. So for example, all occurrences of AG in the graph and also in the multiple sequence alignment, they are related to a specific MSA column. And this gives the conservative approach to uniqueness. And in literature, we already had a linear time construction based on suffix trees, finding all these unique fragments. And also I have a linear time construction of the segmentation, trying to get segments and sequences that are as short as possible. And our contribution for this project is that we consider a partitioning scheme in which we can only pre-process few multiple sequence alignment rows at a time. We can find the corresponding segmentation, and if the corresponding segmentation is not actually unique, is not indexable, then we can just tweak it to make it indexable with just a few tweaks. And this turns out to be at least effective strategy. So we constructed an IFG out of chromosome 22 out of the 1000 genome project variations. So starting from a VCF file. And as you can see, there is some order of magnitude difference between just building the graph containing the variations. So 11 minutes against actually spelling out each haplotype and then finding the unique segments and then segmenting it. And indeed, this was the most embarrassing part of our pipeline, but I spent earlier this month rewriting this founder block graph too. So we got it down at least with the memory and time a little bit. And this partitioning scheme actually makes it possible to run just a few threads at a time and also cut down on space as well. Yeah, but the results are that actually chromosome 22 is made of a lot of short and unique sequences, right? This N50 metric is 18, which means that 18% of the genomic 50% of the genomic content of the graph is contained in fragments in nodes of length at most 18, and this is actually a good metric if we want to do seeding based on unique sequences. And indeed, this segmentation offers less compression than compared to VG, so 68% more base pairs, but also much less recombination, right? Because the VG graph has a gajillion possible paths that you can spell in the full chromosome, but this conservative recombination approach actually cuts down on the number of paths. And right, but after we have built this graph, we want to know how to use it. And the cool thing about the graph is that we can have a fast, exact pattern matching solution. And without going into all the details, the property is that full edge labels, when we see one unique string coming from a certain column and another unique string coming from another column, we can only be in one location in the graph. And this gives us actually a strong property to index. And the indexing algorithm consists of just spelling out all the edges of the graph and indexing this text collection, and then applying this three-step process where we find the largest suffix of our pattern, that is prefix of some edge. And then we actually can chain full edges, and we will have this uniqueness property that guarantees us that we are in only one position in the graph. And then that we have this final connecting vertex step, which for the sake of the implementation, we didn't implement this in linear time, but it was fast on the average case. And our contribution for the paper is that we developed this specific algorithm, and we mainly use an FM index we mainly use text indexing approach. So we require only few basic operations on the text. And how can we get from matching to seeding? Because they are different problems. And we just define this very simple seed, which is a semi-repeat free seed, is any match that spans a full node label of the founder graph. So in this case, the partial match that is in red here is an SRF seed because it spans, it touches, it considers completely a full node label, and that full node label is unique by construction, so it's a good seed. And indeed, we can greedily compute SRF seeds in this way by consuming the pattern until failure. And if we find some SRF seed, then we report them because they are interesting. And if we don't find any, because there are many occurrences in the graph, then we don't report anything. So this is a sort of a very cheap, unique seed that we can get. We can also get long seeds this way because we can optionally report the seeds of the longest, let's say 50 intervals that we matched in this search. and this will also be a good candidate for seeds. And one last step of the pipeline is that we can optionally chain the seeds, and briefly, collinear chaining is this problem of finding the best ordered subset of seeds that is coherent with an alignment, but this time we have a graph instead of two sequences. So we actually relax the founder graph into an elastic degenerate string, and this means just relaxing, allowing all the connections between the blocks. This is still a graph divided in blocks. And then we adapt this chainx algorithm from sequences to this new sequence to graph case. And this chaining formulation is cool because overlap and gap costs give this nice connection, theoretical connection to unit cost edit distance. And the algorithm is actually fast in practice and has a nice time complexity. Okay, so what about the alignment accuracy results? Does this seed set actually improve accuracy? And we use the evaluation pipeline by GraphChainer, which is fixing computing a random path and then sampling reads from them. And these are the alignment accuracy and performance on the normal VG graph. And the optimal one was Graph Aligner at 95%. But by using this new seed set on our founder graph, then we actually get 97.25 accuracy. So we actually can improve the accuracy. And this is the time also shows that this is a good trade-off. This is a better trade-off in time versus accuracy for the seed set. And by looking at the computational statistic, you can show that there is this correlation between the number of seeds that you are considering, so the number of candidate alignment spots where you can align, and also the time complexity. So having a high quality and small seed set is actually very important. And the benefits of chaining, so SRF Chainer is the workflow with the chainer applied. For now cuts down a lot of space, but not a lot of accuracy. So this accuracy actually drops because we are missing some seeds that were corresponding to good alignments. And yeah, so to conclude, we can actually build indexable graphs at chromosome scale, and we showed how to obtain a fast, high quality small seed set. And the limitations right now is that EFG locate this indexing can only index elastic founder graphs or even a larger subset of graphs that has unique nodes. And we do not, in the construction right now, we do not handle large MSA gaps, but that can be studied in the future. And maybe one interesting comparison is with VG because they also build a pattern matching index, and maybe the same seeding strategy can just be applied to their exact pattern matching index. And indeed, all the state-of-the-art tools are based on minimizer seeds, and like in this case, minimizer seeds, they represent too large of a seed set, then gets computationally expensive to compute. Okay, thank you very much for the attention.
Thank you. So we have time for some questions. And maybe I will start with some. So you somehow limit the recombination, but did you somehow look whether your recombination points are ones that are biologically meaningful?
Right, and I think that our approach is principled just because we trust the multiple sequence alignment, and the meaningful biology. We didn't look at any specific example, but we are imposing this unique fragment property. So I wouldn't trust these recombinations 100%, but they look they have a basis in uniqueness.
Mm-hmm. I think we have a question at the microphone too.
Yes. Nice talk. There is maybe something I didn't completely understand, but your technique is really based on an MSA.
Mm-hmm.
And then you compare to other tools which are not strictly based on MSA. If you take Graph Aligner or things like
right.
that.
Yes.
Could you maybe elaborate on this limitation? If it's
right, and of course the limitation is that our approach works only with these kinds of graphs, whether the other aligners are generic and they actually try to align to all the other graphs, and yeah, so we are cheating just because we spend a lot of time in the construction, but this actually gives some better trade-off afterwards.
Okay, thank you.
Okay, so it seems there are no more questions. So let's thank the speaker again.